you for the organizer for the opportunity to present our work. Aaron gave a very nice introduction to the kind of consorting project we're working with, also the technology. So I will just be brief on that. So like the Project HOPE, we are also involved in NCI-funded project using single-cell technology to map cancer tissues. So that's the Human Atlas Network. So the overall goal of this H10 project is really use a range of single-cell sequencing and spatial omics data to really generate reference maps for both adult and pediatric cancers. So our center is involved in Atlas effort for pediatric cancer, three types of high-risk pediatric cancer, high-grade glioma, high-risk neuroblastoma, and very high-risk B-cell lymphoblastic leukemia. So we focus on two critical transitions for all three types of cancer, which is between diagnosis and post-therapy. We use a range of technologies shown here to profile those patient samples. So the overall goal is to really better understand the heterogeneity and the microenvironment of all these cancer types. So for, like I mentioned, one of the cancer type we study is a pediatric high-grade glioma. So we focus on the following three types of high-grade glioma based on the recent WHO classification. Diffuse midline glioma with H3K27 mutation, diffuse hemispheric glioma with H3G34 mutation, and H3 wild type IDH1 wild type high-grade glioma. So we work closely with the CBTN team and our colleagues and CHOP, and we assembled this cohort of 16 patients. For each patient, we have biospecimen from the initial diagnosis, so that's treatment naive. We also have one or two time point from either progression or recurrence, either for the progress biospecimen, either from autopsy or from non-autopsy collection. So for all these samples, we run them through using three assays, single nuclear RNA-seq. For that, for each biospecimen, we actually profile two regions from the same biospecimen to really better understand the heterogeneity within the sample. And we also profile the sample using single nuclear ATAC-seq just for one region. And we generate a high coverage whole genome sequencing data to better understand the clonal evolution. So of all these samples, as you can see here is a number of different molecular class. We have very small number of H3 histone mutation samples and one IDH1 mutation sample, and the rest of them are all H3 wild type and IDH1 wild type cancers. This one shows you the type of the distribution of the single nuclear RNA-seq data across these 64 data sets. So here is the cell type composition based on our RNA-seq data. On the left, you can see it's a UMAP depicting the type of cells we captured from our cohort. Overall, after QC, we sequenced about 404,000 cells. Capture almost most of the major cell types within the tumor. As you can see, the major cluster is the neuroglial cluster in the middle. We also capture some normal, healthy oligodendrocytes or neurons. The largest immune cell type, as expected, is the macrophage microglia population. followed by T cell. We also captured the vasculature, including the endothelium cell and the cells involved in the blood-brain barrier, such as the VLMC cell and parasites. We don't see a big difference in terms of cell type composition across the different time points, as you can see on the right. More or less, the neuroglia cluster remain the dominant number of cells in these tumor samples. This is UMAP based on our QC ATAC-seq data. Overall, we captured about 118,000 cells. Again, the predominant cells are the neuroglia cell, and then macrophage and microglia constitute the major immune cell types. We also capture vasculature and the blood-brain barrier cells. So again, just like the RNA-seq data, the cell composition didn't change much between the initial diagnosis sample and the post-therapy sample, as shown on the right. So for a single cell analysis, it's very important to really distinguish the malignant cell from the healthy cell. As you can see here, even though this big neuroglia cell actually it consists, although based on the transcriptome, these cells are very similar, but actually they consist both malignant cell and healthy cell. So we spend a lot of effort to try to accurately annotate the malignant cell. We use a range of criteria to do this. The major ones are, first, based on the RNA-seq data, we examine the copy number variation pattern of these cells, and then we cross-reference the CNV inference with our deep WGS data about 80x coverage. Based on that, we annotate malignant cells from healthy cell, and we did additional filtering using known marker gene for the malignant cell. So in the end, here are the showing you the different so the annotation for malignant cell and healthy cell. As you can see, even for the neuroglia cluster, the malignant cell has some resemblance, quite a bit of resemblance to the normal cells in this cluster, including the neuronal cell and the oligodendrocyte cell. So like all cancer cells, they are very confused cell. This is especially so for pediatric cancer. On one end, they are driven by all the developmental programs to further develop. And on the other end, they're driven by all these oncogenic events to either evade therapy or proliferate out of control. So this concept recently, this concept of cell state has been very helpful to conceptualize to understand the heterogeneity of different tumor types. So in this analysis, Neftall et al. They sequenced 28 glioma, high-grade glioma samples, most of which are adult samples. Based on this data set and meta-gene analysis, they identified 4 cell states shown on the right: the OPC, oligodendrocyte precursor, NPC, neuroprogenitor-like, astrocyte, AC, and the mesenchymal-like, which are both the 2 previous speakers have touched upon. So these mesenchymal-like cell states really associate aggressive therapy response. So then the question we ask is, how relevant are these cell state and transcriptional signatures to a pediatric high-grade glioma? As I mentioned, the previous cohorts mostly adult high-grade glioma. And then the next question is, can these cell state help us understand therapy resistant response? So then we focus on this central neuroglia cluster, in particularly the neoplastic cells we annotated. So we project our data using the Neftall state score onto this UMAP. As you can see, the answer is to some extent the NIFTEL state indeed can stratify the different cell state to some extent, especially for the NPC-like state and the mesenchymal-like state. As you can see, they form distinct cell clusters. But for the other region of the UMAP, the cell states are largely mixed, suggesting they are not sufficient to segregate all the cell states in pediatric high-grade glioma tumor cells. Here is just showing you the stratification of different cell state in our data in our cohort over time. As you can see, it's the tumor cells are highly heterogeneous in terms of these cell states. Then we dig deeper into this question to look at the patient level and even at a different time point within the same patient, how these cell states change. On the right, you can see again even within the same patient, there's a large heterogeneity between the patient in terms of these cell states. And if you look at within the same patient at the 2 time points, these cell states also shows very dynamic plastic behavior. So, for instance, based on the right, you can see there is a trend actually after therapy. The mesenchymal-like cell states tend to increase based on our cohort 16 patients. So as I mentioned, pediatric cancer, high-grade glioma has a very strong developmental component for these cells. So we reasoned this might not be fully captured in the previous NIFTEL state model. So fortunately, there is a new publication from 2020 from Couturier et al. They generate a reference normal fetal brain single cell RNA-seq data from 4 fetus from the gestation weeks of 13 to 21. As you can see, the reference trajectory captured a range of developmental stage of these glia and neuronal cells. These radial glia cells supposed to be the progenitor cell for these glia lineage, and also glia progenitor cells and oligolineage cells. So using this reference trajectory, we assess the developmental stage lineage of our data. As you can see here, our cohort cells project to different developmental stage, including radial glia, different type of radial glia, glia progenitor cells, astrocyte, and also more mature excitatory neuron cells. So combining the NIFTEL state signature with this developmental lineage signature, we come up with this new 7 cell state model for our PHGG cohort. We still have 2 mesenchymal-like state, astrocyte-like, 2 astrocyte-like state. So the OPC, NPC-like state is hard to distinguish in our cohort, and we also have a more differentiated neuronal cell-like state and also a cycling state. So, one of our main focus is really understand the gene pathways associated with treatment response, because we have such a small cohort and the patient cohort are pretty heterogeneous. So we want to develop a more statistically robust model to identify differential expression. So we came up with this mixture effect model to capture on one hand the random effect from different patients and also the treatment modality effect using the fixed effect model. So using this generalized linear model, we identified about 2,000 differential expression after treatment compared to the initial diagnosis samples. 1,000 genes downregulated, about 900 genes upregulated. So we're still analyzing these differential expression. So our initial analysis, pathway analysis using the GSCA 50 cancer hallmark pathway revealed that there are four pathways enriched in our differential express gene. These include the UV response hallmark genes and interferon response genes. Both are type 1 and type 2 interferon response gene, and KRAS signaling genes, and finally estrogen response genes. All of these pathway genes have been, to some extent, being implicated in glioblastoma progression or therapy response before. But we identified many novel genes in these generally implicated pathways. So we're still following up with these different signature genes. This one shows you the enriched interferon gamma alpha response pathway genes, as you can see here. This is the interferon gamma response pathway genes. Many of these genes are involved in interferon gamma response. And then the next question we are very interested in is to identify the transcriptional regulator of these different cell states, because then we can, these regulator might be a better target to really reverse these cell states during after therapy. So for this, we take advantage of our ATAC-seq data and combine that with our RNA-seq data. So basically, here's the way we try to identify those key regulators. For each regulator, using our RNA-seq data, we can evaluate its activity across the genome using our ATAC-seq data, and then we correlate that these regulon activity across the cell with the cell state signature for the different cell states. So here's the result of our analysis. This heat map shows you the set of key transcription regulators associated with the 6 or 7 cell states.
I'm sorry, can you try and summarize pretty quickly? We're pretty far behind.
Yeah, as you can see, a lot of these factors actually are implicated in Aaron's talk as well, including AP1 factors associated with the mesenchymal-like, anti-associated with the OPC, NPC-like state, and also the TCF4T, the more associated with the developmental lineage, such as for LPC and NPC. So just to summarize, we generate a matched RNA-seq, ATAC-seq, and whole genome sequencing data set for 16 PHG patients, significantly expanded the number of cells to date. And we defined 7 cell states among the malignant cell based on the combination of developmental lineage and differentiation and proliferation states. We identified key transcriptional factors regulating each malignant cell states. We found that interferon response pathways significantly enriched in post-therapy samples. With that, I'd like to thank our village to generate this kind of data, and for the research, people in my lab, Qiahui Chen, Yusha Sun, Jonathan Sussman, Anusha, and Wenbao, and our collaborators, and CBTN Biobank, and also patient and family. And thank you.